I'm Alan and welcome to this video. The title of the presentation today is Investigating Functions of Hypervariable and Hypovariable Genes Using RNA-seq Data. This is a summary. So expression variability, or otherwise called EV, is a metric which measures the degree of variation of gene expression, and it is magnitude independent. Now I'm going to be explaining this more in detail in the later slides. And the EV is actually influenced by the epigenome. So EV has been repeatedly reported to be associated with the methylation disease and aging, so it is important. And in this study, we found 2,730 hyper and 545 hypovariable genes based on the EV matrix. And hypervariable did not have any significant enriched function, whereas the hypovariable genes were had the conserved and essential cellular processes such as cell cycle. So this is the main figure, and we can see the slides are the figures are explaining the functional enrichment of EV genes. The x-axis is the full enrichment or the importance, and the y-axis is the function itself. And we can see for the hyper, there's no specific enrichment except for the protein digestion. But now we've actually looked at the top genes of hypervariability, and they were HLA and EIF1AY, which are involved in immunity and sex specificity. So we know that in the human population, the people differs in their expression significantly in terms of the immunity and the sex. Whereas on the right side is the hypovariable genes, and we can see the transportation, histone modification, RNA splicing, and cell cycle, which are all really essential cellular processes, are actually hypovariable, which means that in the human population, there's really not much variation for this specific kind of mechanisms in terms of the gene expression. Now, in terms of the expression variability, we've actually used expression variability because it's magnitude independent, and variance itself depends on the magnitude of expression. So variance is actually biased towards the highly expressed genes because, as you can see on the right side, the magnitude is proportional to the variability. So the higher the gene expression, as you can see over here, the higher the variability or the variance. So we cannot actually use this matrix to fairly compare between the genes. And there's another matrix called coefficient of variation, which actually adjusts by the mean. But this is two way to conserve and construct. It is biased towards the lowly expressed genes. So we've actually chose to do this experiment with magnitude independent bias-free matrix called expression variability. This is how you actually calculate it. We've actually used the non-parametric version of the calculation. So expression variability is essentially the observed MAD minus expected MAD. Now MAD is median absolute deviation, and it's calculated as the following equation. So what we do is to actually get the deviance from every gene expression and find the median from that. So this is essentially the same or similar to the variance or the standard deviation, but we're doing this by the median, which is non-parametric. And what we do is as to how we find the expected MAD is what we do is to do a local polynomial likelihood estimation. And this is also called the low-West smoothing. And this is the figure which shows you that. So this is the expected MAD after fitting the local polynomial regression or likelihoods, and this is expected, and we compare that to the observed MAD. So the residual or the deviation of these two is actually the expression variability, and on the downside is the actual EV, and EV should be supposed to be 0 if the expected is the same as the observed. So this is our criteria for getting the hyper and hypovariability genes. So this is the total frequency of the genes within a human sample, and we can see that most of the genes are actually non-variable at around 0 expression variability. But importantly, there's some genes on the left side and the right side that are really variable or really not variable. And so on the left side, which is around the median minus 3 MAD, is the hypovariable, and the right side is median plus 3 MAD, which on the right side, and we've actually chosen by this criteria around 2,000 genes over here and 500 genes around here. So that was it. And we want to actually acknowledge by the references that the calculations were derived from another sources, such as the determination of expression variability by the Alemu et al. Sorry for the pronunciation. And the exact MAD or non-parametric version was introduced by the Beshkeel, sorry for the pronunciation again, Beshkeel et al. In 2019. Yeah, thank you.